We can help introduce you to what Twist can do for you. And with that, I will hand it over to Guannan. There.
Thank you, Jason, for the introduction. First, I'd like to thank Twist Bioscience to invite me here to talk about my work, but most important of all, to give me this opportunity to walk so much every day since last Friday. So it's very healthy. And so on the menu today, this is my disclosure. This is a Twist-sponsored presentation. So on the menu today, first I'd like to briefly introduce to you the concept of precision medicine and how it is changing the landscape of human oncology. Of course, by now, everyone should be very familiar with the concept. And next, I'd like to share with you our effort to bringing precision medicine into veterinary oncology, applying NGS-based diagnostic test in canine hemangiosarcoma to identify clinically actionable biomarkers. And lastly, I'd like to talk to you about our effort to build a comprehensive canine cancer panel designed to use in many common canine cancers. So I'd like to start today's seminar with a sad story about a 7-year-old German Shepherd dog named Jacob. Jacob was the happiest dog in the world. And one day, so he started to show lethargy and weakness before he completely collapsed a week after. The ultrasound showed hemoabdomen and a big splenic mass, which was later diagnosed as hemangiosarcoma. So the primary tumor was removed right away and followed by chemotherapy to slow the metastatic spread. But 3 months after diagnosis, Jacob passed away due to metastatic disease to his lung. And this is one of the sad stories about a million dogs that are diagnosed with hemangiosarcoma, and nothing we can do at this moment. So being a hardcore dog lover who have witnessed the rise of precision medicine and how it is revolutionized cancer care for human oncology patients by delivering the most effective treatment based on deep patient phenotyping and genotyping. So Dr. David Ross and I have the same goal to bring this concept into veterinary medicine. With the help of 2 great veterinary physician scientists, so we set to test the concept of precision medicine in the cancer that Jacob had and so many other dogs had, canine hemangiosarcoma. It's a aggressive malignant sarcoma, and it's one of the most common canine cancers in the dogs, and it progresses rapidly. Most dogs died within 6 months due to metastatic spread to their lungs, and so it does have a certain predisposition. It's overrepresented in German Shepherd, Golden Retrievers, and Labrador Retrievers. So these features make HSA an excellent disease for us to pioneer precision medicine, such that there is a readily available tissues for us to do any genotyping, phenotyping we want to do, which is not the case in some of the human cancers. And it's 100% desperate and fatal, and it has a strong human correlate because there is an equivalent human sarcoma called angiosarcoma. It's equally desperate, and it's very rare to even study it. So we hypothesized that if we assess tumor mutation profiles with next generation sequencing, we should be able to identify oncogenic drivers and clinically actionable targets which hopefully will advance therapy and improve outcome for this desperate disease. And to do that, I first performed whole exome sequencing to investigate the mutation landscape of canine HSC and identified oncogenic drivers and biomarkers that looks like they have prognostic or therapeutic values. Next, I built a targeted resequencing panel, we call it the HSA panel, to validate these biomarkers that I identified from the discovery sequencing in a larger cohort, and also explore the possibility of using this panel sequencing as a diagnostic test in veterinary for veterinary patients. And last, we want to evaluate these biomarkers in larger prospective studies. So our exome workflow is very straightforward. So except that we did use canine all exome capture solution instead of human, and I did sequence both normal and the tumor and paired normal samples to detect true somatic mutations. The annotation and the interpretation of the results, however, is pretty challenging because, as we all know, that canine cancer database is just it's very limited or not available at all. So that's why I had to rely heavily on human database to predict and understand the function of the mutated genes, the pathways that are involved, and also the most important of all, the implication of these mutations in cancer. What does it do in canine cancer? So a lot of stratification from human database. And our customer canine bioinformatics pipeline allowed us to identify oncogenic drivers and to understand their biological and clinical implications. So together, our canine sequencing, our exome sequencing identified oncogenic drivers in more than half the cases that we sequenced, which is 20 HSA cases. And so these cancer genes, out of the 4 cancer genes that we identified, 2 of them are oncogenes, PIK3CA and PLC gamma 1, and 2 of them are tumor suppressor genes, P10 and P53. So several points I want to make here. Not only are the genes well-established cancer genes, the mutations we found within these genes make a lot of sense. They are well-documented oncogenic activating or inactivating mutations. So such as PIK3CA, PIK3CA H1047R mutation. This is one of the most common enzymatic activate mutant for PIK3CA. And it's almost the most common mutations in human cancer. And PLC gamma 1 S273F, this corresponding to PLC gamma 1 S345-F mutation, which is another common enzymatic active mutation in human cancer, including human angiosarcoma. So this is the first evidence that we found, similar mutations, equivalent mutations in both human and canine. And of course, loss of function mutations in P10 and loss of function mutations in P53. So we were very excited about the exome sequencing, but we can't ignore the fact that exome sequencing failed to identify driver mutations or markers in almost another half of the cases that we sequenced. So based on several lines of evidence that I hypothesized, it may be due to that whole exome sequencing is lacking the sensitivity that we are looking for in cancer to identify mutations in cancer. So we probably need a more sensitive approach. That's when we decide to design a targeted resequencing panel, which focuses on a limited number of genes that are potentially involved in HSC tumor genesis. And a lot of the design and data is of course based on our exome sequencing data as well as genomic data in human angiosarcoma. So for method I chose to use the amplicon-based sequencing. This is a perfect method for a panel this size, around 60 kb, and it's also compatible to use with FFPE samples. So this is our HSA panel. It covers 4 coding regions of 24 genes and hotspot mutations in another 7. So it includes recurrently oncogenic driver mutations, recurrently mutated genes, and hotspot mutations. And I also throw in a few common cancer genes in both human and canine solid tumors, just to increase the the clinical utility of this panel. And our panel sequencing, reproduced the exome findings by re-identifying the same mutations in the same cases. But it also make new discoveries. It discovered potential candidate driver genes in another 8 cases, as indicated by the triangles here. So in the additional cases, not only do we see the same 4 cancer genes, I also found a recurrent unrest mutation in another 3 cases. So it's exactly the same. And the unrest mutation, I realized later that the allele frequency is relatively low, may explain why we missed it from the exome sequencing. Again, it's highlighting the power of the panel sequencing. So the significance of this unrest, so all 3 unrest mutant are Q61-R mutation. So in the human equivalent Q61-R, this is a well-known activating mutation. It's a driver in many cancer types, such as melanoma, lung cancer, but it's also found in angiosarcoma. Unrest is upstream of MAC kinase pathway, and so there are multiple inhibitors, inhibiting this pathway. So this is a very attractive drug target. This is another, evidence that we found mutations shared between canine HSA and human AS, suggesting, strong human correlate. And the fact that, we missed it from the whole exome, so I actually went back to see if it's indeed there. They just got filtered out by all the, filter steps during the whole exome sequencing. So then we employed the panel, the HSA panel, to sequence additional 30 HSA cases. So including the first 20, so all 50 cases are summarized here. So we found the same candidate driver mutations, NRAS, PIK3CA, PTEN, P53, and PLC gamma 1. Now we see a pattern. So NRAS is present in 12 out of 50 cases. Majority of them are lacking P53 co-mutations with a few exceptions. And PIK3CA, 24 cases bear PIK3CA mutations, and 3 have PTEN mutations. Most of them do have P53 co-mutations. And remember, so PIK3CA and PTEN, they are key components of PI3K signaling. They are very upstream. And the PTEN inactivating mutation works in parallel with PIK3CA activating mutation. And they both elevate the PI3K signaling and also very attractive drug targets. And in 2 cases, we found PLC gamma 1 mutation. Both mutations are active mutations. And so out of the 50 cases, I didn't find any legit drivers in 4 cases here, which is none. And in another 6 cases, I found P53 mutations only, no other major drivers. So, but overall, so it's, the HSA panel is informative for more than 90% of the HSA cases that we tested, suggesting high diagnostic utility. And if you note that, all the major driver groups, they are mutually exclusive. There is no overlapping between them. So this might suggest that, these driver mutations might represent molecular subtypes of the HSA that was previously thought as a single entity disease, as shown here by this, histology image. Under the microscope, we think it's a single disease, but now it actually consists of multiple molecular subtypes. And 2 subtypes, actually 3, NRAS, PLC gamma 1, and a few P53 cases. So they are shared between canine and human, suggesting that although genetically heterogeneous, a subset of canine HSA might bear, similar genetic driver mechanism as their human counterpart, and suggesting that canine might be able to, we might be able to use it as a model for human AS. And the NRAS and the P53 group, so this account for more than 75% of the cases that we tested, and so both of them are attractive druggable targets, suitable for pharmacological intervention. There are both FDA-approved drugs or investigational drugs available to target these pathways. And we are fairly excited about the genetic findings, but my question remains: does the activated upstream mutations lead to elevated downstream signaling in canine? So where they know this in human, does it hold the same in canine? So indeed, the IHC staining showed beautiful staining signal of phospho ERK, which is a classic marker of MAP kinase pathways, suggesting that NRAS is indeed related to downstream activated signal, and a direct evidence that NRAS mutations activate, drives tumor genesis in these cases. Similarly, the activation of PI3K signaling is also confirmed by the expression of both phospho AKT and phospho S6, 2 downstream effectors of PI3K signaling. So at the end of, the functional results is actually pretty gratifying, since it's consistent with previous findings that these 2 pathways are indeed activated in a subset of canine HSA cases. Now our mutation profiling actually provide a genetic mechanism for these phenotypes. So by the end of this process, so we had in hand a functional relevant, cost-effective test, which could be can be used for archived cases of HSA for research purposes, but also have the potential to screen, standard veterinary patients in routine clinical care. So I'm listing here a few directions and projects that are inspired by this work. But there are many other aspects of canine HSA that we didn't, that were unthinkable before this. So in the interest of time, I'm just gonna show you how the genomic findings and HSA panel powered the design and the development of a precision clinical trial. So providing not 1, but 2 options. In one scenario, so after the genomic profiling present druggable targets, and based on the data I have so far, 50% of the HSA cases will have PI3K related mutants, and they are eligible for PI3K inhibitor trials, and 25% cases have NRAS mutants unRAS mutants, and we can enroll them into a MAP inhibitor trial. So of course, finding affordable drugs is a whole other story, so I don't even want to get into that. But fortunately, our genomic findings present an alternative option by taking advantage of the extremely constrained mutation landscape of HSA and design listeria vector-based immunotherapy therapy clinical trial. So this work is currently underway in collaboration with the company Bioscience, where we can take the HSA-specific mutations and stick it into the listeria vectors and make a off-the-shelf vaccine to treat HSA patients that carry these mutations. Of course, we will still do the screening upfront and screening after treatment. And so this is, of course, one of the examples of the many advancements that HSA panel can make. And a perfect segue to move into the last section of my talk. The long-term goal of our collaborative effort is to really bring precision medicine into veterinary medicine, veterinary oncology. So based on the rich experience we gathered in the HSA work, so I now turn my effort into building a comprehensive canine cancer panel. So this panel is detect a wide range of mutations in 284 genes known to play a role to drive cancer mutation, and covers a total target region of 1.4 megabases in canine genome, and that's almost 4% of the exome, canine exome. And so I chose to use hybrid capture-based enrichment sequencing, and this work is being done by Twist. And so our goal is, of course, to map mutation landscape with this large, broad panel and identify driver mutations and clinically actionable markers to arm clinicians with better prognostic information, more therapeutic options, and of course provide markers that might predict treatment response, as we did in human. So our canine cancer, our broad cancer panel covers the genes in the key signaling pathways known to play a role in cancer, such as Wnt signaling, p53, and epigenetic, Notch. And it also, the design is multifold. It also covers all the potential drivers in the most common canine cancers, the top 10 canine cancers. So, we are very excited about this, about the panel, and to the best of our knowledge, this is by far the largest and most comprehensive cancer gene sequencing panel in veterinary medicine. With every great opportunity comes with decent amount of challenges. So I want to show you here a few lessons that we've learned during this process. So first of all is the design of the probes. So it's you would think it's an easy switch between human genome to canine genome. It actually requires serious coding changes and informatics resources. In the end, Twist basically had to rebuild the whole design pipeline to 4 non-human genomes, including canine, and I know for a fact that they also tested in mouse. So hopefully things will, the design will go much smoother in the future for all non-human projects. And so another thing is that it's a common practice to include blocking agent in the hybridization reactions. So this is to remove the non-specific binding associated with repetitive sequences in the genome. And human COT1, so I think most people have heard of it, is such blocking agent. It's enriched with repetitive non-coding elements. So we just thought, okay, we're gonna use human COT1 in our canine reactions. and hopefully we'll do the same with canine samples. But our on-target rate actually really suffered before we realized that, there's just not enough sequence homology between human genome and canine genome for that to work. So moving forward, we'd like to use a canine-specific blocking agent, and so hopefully this is a lesson. This is a lesson for us and hopefully for others as well. So currently the design and development is several rounds of them have been completed, and so the validation is underway, and the initial matrix looks really good with a very high uniformity and a very decent duplication rate. So we're just right now working out a few kinks to improve on target. And so I'm using for validation sample, I'm using my HSA cohort. That's a no-brainer. And so glad to find out that it, basically reproduces all the results that we found before, and it made new discoveries. So we figured that it might be due to the fact that the capture enrichment method is more tolerable for poor DNA quality than the amplicon-based method. So remember, all of my samples are FFPE samples. So we were pretty happy to get to where we are now, and so this provide a model between the discovery, the exome sequencing, the focused small panel, and the broad large panel. We achieved almost 100% diagnostic utility for canine HSA, and this is a model that I'd like to introduce to other canine cancers as well as other species as well. So to summarize our effort to bring precision medicine into canine HSA, the genomic profiling and the HSA panel allowed us to find driver mutations, to better understand the biology and the pathogenesis of canine HSA. We also demonstrate that HSA consists of multiple molecular subtypes. Some of them have a strong human correlate, and the druggable target subtypes. So they helped design the precision clinical trials so far, which hopefully will provide at least novel strategies for us to try and transform how HSA is treated. And hopefully this will lead to tailored, more effective treatment therapy. Another thing is that our working in canine hemangiosarcoma serves as a proof of concept study to introduce precision medicine in other canine cancers as well. It already led to the development of our comprehensive canine cancer panel. And so we're hoping that our work here is not only valuable for veterinary patients, for canine patients, we may also be able to translate the canine the results in the dogs to human diseases, especially those human diseases that are too rare to study to mount a meaningful clinical trials, not to mention to find an effective cure. I think in the end, this is a win-win situation for both us and our canine companions. So with that, this is my founding sources, and this is our fabulous group of people that made this work possible. So I'm currently actively seeking collaborations. So anyone who are interested in certain canine cancers or have access to canine tumors, feel free to reach out. Thank you.